I think we're live.
Awesome.
And so what this is going to open up a YouTube page, and so you can just close that down when it
Mhm.
opens up, so that there's no echo and such.
Cool.
Yep, we can see it. Everything's great. We are
All
live,
right.
so you may begin whenever.
Awesome. Well, welcome everybody. So today I want to talk about um, AI designing drugs, which is a bit of a controversial topic, but yeah, hopefully we can break it down. And um, I guess to start off with, I would like to introduce myself, tell you a bit more. So I'm actually, to my understanding, most of the audience is students of the Edinburgh Uni. So I'm actually an alumni. I graduated last year. Um, and overall, I really enjoyed my experience at the University of Edinburgh. So I hope you're having a good time too. I studied artificial intelligence and computer science. And just to give you like a bit of a breakdown of what I've been up to like recently. Um, so after my second year, I did an internship at JP Morgan, which I don't know if it's like still very popular among computer science students, but at least in my year, it used to be like extremely popular to go to JP. A lot of graduates are there now. Yeah, it was a really interesting experience, and it was like pretty cool because a lot of my classmates were doing the same internship. But in terms of like the project that I was working on and just sort of like the tasks that you encounter in banking, that wasn't all that exciting to me. So like socially, great experience, sort of like technical challenges. I enjoyed that part slightly less. And then in my third year, I did an exchange and I went to Caltech, and that was really fun. That was pretty challenging as well. But yeah, it was like a pretty eye-opening experience in a sense that it was like a first year where I was exposed a bit more to research and how research works because Caltech is way smaller than the Edinburgh Uni. So you actually can get a hand of your professors pretty easily, and you can like interact with them and just chat about like what type of research do you do and like what stuff are you interested in, and you also interact a lot more with like PhD students. So there's a lot of like research talk in general going on, and a lot of people want to like pursue research after they graduate from Caltech. So yeah, for me that was really cool that I got to both like learn a bit more about like what type of research is happening in computer science. And it was also interesting because I got to see like more hands-on about like what research, yeah, how to do research and like how do people approach it in general. So then after that year, after the school year finished, there is this program at Caltech, which is called SURF. It stands for Summer Undergraduate Research Fellowship. Um, and actually, if any of you would be interested, you don't have to be a Caltech student to apply for that program. So it works like an internship. You get paid for doing it. And people from like all sorts of different places apply. And then you get to pick your professor who is going to be like your mentor or your supervisor during the project. And then you get to do a research project for 10 weeks. So I did that with like a machine learning professor. His name is Yi Song Yu, and yeah, and the project was on multi-agent reinforcement learning. And the problem itself was really interesting, although it was a bit challenging. And by the end of the summer, I had some results, but by no means it was like a finished project. So I actually decided to carry on with working on the same problem, and then I basically made it my dissertation project in my fourth year uh, at Edinburgh Uni. So but I had like a different supervisor from Edinburgh. Yeah, so that was my 4th year. So yeah, as I said, I graduated last year. And then from then on, right after graduating, I went to Singapore, and I've been there for a couple of months. And there's this research institute which is called A*STAR. So I've spent a couple of months there, and then I was working on also reinforcement learning. I'm genuinely pretty interested in reinforcement learning. This is the area that I'm like most passionate about. So I was working on a reinforcement learning project as well, but very different problem where you're trying to apply reinforcement learning to when you have like those really, really large neural networks that have a lot of parameters in them. You're trying to apply reinforcement learning to prune that network out of the unnecessary parameters. And you're interested in pruning grades of like 80, 90%. So really most of the parameters go out of that network, but then you don't want the performance of the network to go down. So you're trying to get rid of basically the unnecessary parameters. And you're to sort of like do it in a smart way, you try to use reinforcement learning to do that. So that was like in the research sense, that was a really cool experience. I unfortunately didn't get to know that many people in Singapore. So yeah, that was a bit so-so, but yeah, the research project was really cool. I really liked that. And that brings me to where I am now. So from January this year, I started working at Exscientia, which is a small startup based in Oxford, in the UK. And yeah, basically what they do is they try to apply AI to design drugs. And my position at Exscientia is called a research engineer. So yeah, I basically do research that is supposed to help out in designing drugs. Yeah, so that's a bit about me. And then in terms of the talk, so the overall idea is that I would like you to, and when I was preparing the slides and sort of like deciding on what I want to cover, basically the goal is that if you walk out from this talk, I would like you to have like a broad overview of what the drug design process is like. And if you were to apply artificial intelligence to it, then like to sort of know like which components of that process maybe you want to target, and maybe like in the very broad sense, how would you go about it? So that's the goal for this talk. So okay, to start off, I want to talk about how drugs are designed in general. So it's actually a very long and a very expensive process. So it makes sense to try to optimize it. And basically what you're going to try to do with AI is to make this process shorter. And through making it shorter, you're also going to basically generate some savings on the financial side as well, because if you don't need to spend as much time on it, then you're not going to need to invest as much money in it. All right, so in terms of the stages of the drug design process, so okay, so you have a disease and you would like to figure out a drug that is going to be able to stop that disease. And well, that's a non-trivial problem. So you break it down into smaller tasks. So well, long ago, it was basically a huge guessing game in terms of like what maybe is going to stop the disease, or maybe it's not going to. But now we have better technologies, and the research is a bit more advanced. So what we can do is, the way diseases work in our bodies, it's that basically there's like a pathway of a disease. And I'm no biologist and no chemist, so this is a wide oversimplification, and yeah, I'm I'm a computer science person, so yeah, it's basically like the way I understand things, and it's not super specific, but basically there are there's like pathways that the diseases take, and if you can break like at least one point in that pathway, then you can stop the disease. And the way like those like the stops on this pathway, they happen through like proteins and different enzymes that like it's basically like a chain reaction. And if you can stop one element, then maybe you can stop the whole disease. So now you're trying to identify those proteins that if you can bond to them with a small molecule, which is a drug, then you engage it in a different reaction, sort of, and then it's not participating in that chain reaction that is causing the disease anymore. And those proteins we call targets. So basically, it means that if I can design a molecule, which then becomes a drug, that is going to bind to that protein in a specific way, then I'm going to stop that chain reaction and therefore the disease from happening in our bodies. So that's the first stage, identifying those targets, those target proteins. And then once that's out of the way, and we're pretty convinced that the targets are actually correct, then we can try to start looking. So the hit discovery phase is where we start looking for molecules that are actually active against our targets, against our protein. And by active, we mean they can bind to the active side of that protein and then engage in some sort of interaction with it. So yeah, we're trying to look for those molecules. And then once we found a group of active molecules for our target, then now we're going to select the most promising ones out of those, that large group of protein, the molecules that we found. So this is the hit to lead phase. And now we have the lead optimization phase. So now we have a small group of promising molecules. And from walking out of the lead optimization phase, we want to have drug candidates. So these are going to go to like animal testing and then human clinical trials. So what we want to do in the lead optimization phase is like take those molecules and just tweak them slightly to make them just better like potential drugs. And then walking out of that lead optimization phase, we have, well, drug candidates. And now we need to test them in order to like convince ourselves that they're actually good drugs. So at first, we test them on animals, usually. It's different types of animals. And then they go off to human clinical trials, which is a very lengthy process. And there's usually 3 phases to it. And each phase involves like a larger group of people. And then after all that is successful, then we can get an approval. And then we can say that we've created a drug for this disease. So it's actually a very long process. And it's pretty complicated. But some of this we can address with AI. And this is both like these stages that I've highlighted in this box. These are the problems that in principle, the AI should be able to be applied to them quite easily. And also, these are the parts that Exscientia, so the company that I work for, is mostly targeting. So we are mostly working in these 3 areas. Okay, so moving on. So if we wanted to design AI that is going to solve those 3 phases, this like hit discovery, hit to lead, and lead optimization, this slide is mostly focusing on the earlier stages. So we mostly think here of like hit discovery and hit to lead. Then in principle, everything should be very simple. So in principle, if I was going to design a drug, well, I would go from the target selection phase, and I would already know the proteins that I want to bind to, that I want my molecule to be active against. And then some molecules, some proteins, they call them off targets. So some proteins that I don't want to bind to. So this might be some protein that is going to like cause some sort of reaction in your brain, or just something bad that you want to prevent because it would mess up with some other processes in our bodies. So we don't want to touch those. So we have a set of targets and we have a set of off targets. And that's basically an objective. So now I have a pretty well-defined problem. I have an activity profile, and I just want to find molecules that basically fit into that activity profile. All right, so what do I do next? Now that I defined the problem well, I could actually use the AI elements. So I could grab a generative model, and we're keeping it like super abstract, super high level. So I could have a generative model, and that generative model is just trying to generate drugs that fit that activity profile that I want. And then to know if they actually fit the activity profile, I would need some sort of predictive model. Because usually, if you wanted to check, if I give you a molecule and I ask you to check whether it's active against a given protein, against a given target, then what you would need to do is you would need to run some tests in a lab. And that takes a long time, obviously. And that takes, like a bunch of people need to get involved. The molecule has to be synthesized, and then you have to do some like actual physical biological tests. And only then you can determine a score. But if I could build a predictive model to do just that, to predict the activity, then I'm saving so much time and so much energy. So now I want to have this interaction. I want to have a generative model that generates molecules, and then I want to have a predictive model that can tell the generative model if it's doing a good job. So the predictive model predicts the activity, and then the generative model is trying to generate better and better molecules. So this here is kind of like a loop going on. And then to get to this sort of like hit to lead phase of the drug design pipeline, now I would need probably some sort of like selective model to select molecules. And then probably to move on to the lead optimization phase, there would probably be like another cycle going on where I choose and then I tweak and then I choose again, but I didn't include this in the diagram just to keep things simple. Yeah, but in principle, the whole thing should be pretty straightforward. Just like grab a generative model, it can be implemented already, just apply it to our domain, then give me a predictive model. Again, it can be already implemented, just apply it to our problem. It's like feed it the right data and make it do its thing. And then everything should work. So if it's this simple, then why is there no AI designed drugs? So we're going to get to that in a second, but just to clarify things, when we talk about predictive models and training predictive models, actually predictive and generative models are pretty similar to each other. They're just trying to learn slightly different things. So if we want to train a predictive model, what we do is we usually take some sort of data. So in this example, you can think of it as like images, and we're just trying to identify whether the images are images of cats or not. So I have some trained data. So this trained data is images together with labels. So the labels tell me, yes, this image has a cat in it, or no, this image doesn't have a cat in it. And all of the data is labeled. So now the model has a lot of examples of like what is the correct answer in a particular case. And now from these examples, so this training data, it's trying to learn. So this is the process, and it doesn't really matter what model you use, what type of predictive model you use. This diagram stands true in general. So it doesn't matter if this is a random forest, a neural network, just like a simple logistic regression. No matter what you use, like this process is basically the same. So this is, depending on your model, the specifics of how you're going to train the model change. But this is some sort of training process happens here, where you train the model using the training data, which is examples where everything is labeled correctly. And this process leaves us with our model. And then to make sure that the model is actually doing a good job, you want to test it on some sort of validation data. So you feed it the validation data, and then the model predicts. So for example, for this particular case, it should tell you that, yep, this is a cat. Or for some other image of a dog, it should tell you, nope, that's not a dog. But you want to check if this is actually the case. So you want to check how good of a job it's doing, and then you want to evaluate it. And then if it's bad, you can go back to this training step and retrain it again, to take into account sort of like that performance. And yeah, hopefully if you keep repeating that through time, your model is going to get better. And then at the end, there should be one more data that you have completely held out. We call that test data. And then at the very end, you might want to evaluate your model on a completely unseen data and see how it's doing to get a better idea of how it's actually performing on something that it has never seen before. And for generative models, you're basically doing the same thing. Like, as you might have noticed, not many elements of this diagram have changed. Apart from here, I don't generate a prediction. I generate a sample. So this data is not going to be labeled. And so if this is a data set of faces, then the model is just trying to learn how to generate faces from examples. So then here, the output of the model is not going to be a prediction, it's going to be a generated sample. So if the model is good, then it's going to be generating faces in this particular case. So then the objective that you're trying to achieve is to generate data that resembles your training data. It's not that training data. You're not just reproducing the same thing. You're generating something that comes from a similar distribution to your training data. So this is just, yeah, again, like very model agnostic, just in general, what I mean when I say predictive model and what I mean when I say generative model, and also how would you go about training them. All right, so now back to the molecule stuff. So the question that I left you off from that drug design pipeline diagram, well, and then the slide of like applying AI to it. The question is, if everything is so simple and it just takes a predictive model and a generative model, and basically you can generate good compounds with that, then why aren't we seeing drugs designed by AI already? And the thing is that actually, if you take a bit of a deeper dive into what those problems actually are, then it turns out that at most of these stages, the stages of training the models, there are some challenges that maybe you wouldn't have think of just at a surface level. So if we take the predictive models example, then from the data side, so you basically, the problems with the data that you encounter with molecules a lot is that a lot of the data sets that are created, so in our case, the problem from the previous slide, in the previous slide, we talked about classifying images of cats. Well, now we want to classify the activity of a certain molecule against a certain target. So that's our predictive model that we are building. So the data that we should have is the activity data of molecules. But the thing is that since that data oftentimes comes from publications, and people tend to report results about active molecules, but not so much inactive molecules, then our data is oftentimes biased towards active molecules. So there's loads of missing entries, loads of stuff that people have never tested before. And then the entries that we do have often tend to be data about active molecules, but not so much about inactive molecules. But we want both. So that's a problem. And then another thing is that since it is data about activity of molecules against proteins, the measurements that are in the data sets, that they had to come from some sort of labs. Someone had to measure it. And the thing is that even if the same measurement was repeated in the same lab, chances are that the value that you would get out of it would differ. And it's even more so the case when you consider two completely different facilities doing some sort of measurement. So the data in our data sets, the molecules data sets, is oftentimes very noisy. So not only it's biased towards just active molecules, but it's also noisy. So your models have to be pretty smart about like training on such like limited data. And then if you try to split the data into train, val, and test sets, there's problems with that too, because sometimes molecules are very, very similar to each other. So if you would have like one of the, let's say that there's two very similar molecules, one of them you end up putting in your train set, and the other one you end up putting in your test set, then even though technically they are slightly different molecules, they're actually so similar then that there is knowledge leakage between the two. So you would want to avoid doing that. So before you even cluster split your data into the 3 train val test sets, you might want to cluster it first so that you have clusters of similar molecules together, and then they all go to the same split. So the same sort of like group of the data, to make sure that the performance that you're measuring out of your model is actually representative of how the model is going to be performing on unseen data. And then with evaluating the models, it's again very unclear sometimes what metrics to use, because it might differ on the application of the model, because you might want to use your predictive model for interacting with a generative model like we had in the previous slide, but you might also want to use it for selecting the molecules like in the hit to lead phase. And then you would want to be looking at slightly different metrics. So yeah, it's pretty complicated. And basically like each stage of training your model, if you start digging deeper, turns out there's a lot of issues that you need to overcome. And that's why those problems are oftentimes very challenging. And that is also why we haven't seen that many molecules generated by AI just yet. All right, so now I wanted to take a little tangent and tell you a bit more about what I personally do and what type of research I do at Exscientia. So as I mentioned at the very beginning, I'm interested in reinforcement learning, and I find it a particularly fascinating sort of like wing of the machine learning methods and AI in general. So just to give you a brief overview of what reinforcement learning is, reinforcement learning is basically this interaction between your agent and your environment. So now you have this loop between the agent and the environment, and the goal is for the agent to learn how to interact in your environment. And in order to do that, it's going to be in a certain state of the environment. So let's think of like a person sitting in the room trying to sip on their coffee. So their goal is to take a sip of coffee. So if they reach for a cup, that's a pretty good action to take if you want to take a sip of coffee. So your current state is just your room that you're sitting in. And then if you take an action in that state to reach for the coffee, that's a pretty good action. And then the environment is sort of like going to update your state, your current state, where now it went from a state where you're just sitting to a state where you're reaching up for your cup. So, and then after you took that action, the environment is also going to tell you how good of an action that was. So how much closer did it bring you to actually achieving your objective? And that thing we call a reward. So if something brings you closer to achieving your objective, supposedly you're going to be getting like a positive reward. If it takes you further from achieving your objective, then you're going to be getting a negative reward. So the environment grants you this information through this reward signal. So now, and that's basically the principle that reinforcement learning is based upon. So in the predictive models case, you usually had a data set that you learn from. In reinforcement learning, you don't start with a data set that you learn from. You learn from this interaction. So you learn through receiving those reward signals, which I think, and the reason why I personally think that it's pretty fascinating is because it's a bit more similar to the way that humans learn. Like if you're a baby and like you touch a hot cup, you get a negative reward because you get burned. So next time you know not to touch something hot. So yeah, that I think that's pretty cool. And I think it's cool that you can also apply it to algorithms, and they can also figure out ways to navigate different environments through the same principle. But yeah, well, it turns out you can apply the same approach to molecules, and you can actually generate molecules using the same approach. So if your current state or situation is the molecule that you have built up so far, and then your action is basically another thing that you're going to add to your molecule, and then the reward signal is how much closer this brought you to this molecule being active, then you've modeled generating molecules as a reinforcement learning problem. So that's what I'm trying to do with my work. And basically, just to give you a slightly better idea of how this works. Again, I don't, I'm keeping everything very sort of like not very detailed, but yeah, let me know if you have any questions at the end, and I'm happy to explain a bit more about the process. But basically how it goes is that we operate on this notion of a policy. So the agent is trying to learn a policy to navigate its environment. And it has some sort of policy to start off with. And it tries picking different actions at different states. So in terms of generating molecules, you start with like an empty molecule that just has sort of like a start token. And another important thing that you should know is that in terms of every molecule can be represented as a string. And those strings for molecules, we call them smiles. It's a specific syntax that allows you to uniquely encode every single molecule as a smile string. And so we operate, to generate those molecules, we operate on those smile strings. So when I say that the algorithm or the agent generates a molecule, in this particular reinforcement learning approach, it means that it's going to be generate smile strings, and it's generating them character by character. So it starts off with an empty string. Then there is some so according to its policy, there's distribution over the actions that it can take. So it basically samples this distribution, and then you pick your next action, so your next token of your smile string based on this distribution. And then the distribution changes because now you're in a different state. So here you are just in the start state, and now you're in a start state together with a carbon atom here. So you've added a carbon atom to your string and to your molecule. And now you're going to sample a slightly different distribution that is conditional on this new state. And you're going to sample another token to add to your smile string. And you're going to keep repeating that process. So keep sampling, 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 sampling up until you sample an end token. So that basically means that I've completed my molecule. Now the agent has constructed a full molecule that, that is trying to optimize a certain objective of, as we said earlier, an activity profile. So now I'm going to evaluate how good of a molecule that was. So think of that in terms of like the reinforcement learning framework, that's the reward. So we're going to grant the agent a reward, and that reward is also the activity of the molecule. So does it fit into the desired activity profile or not? And if it does, then I want to update my policy so that those probability distributions in a way where sampling a similar molecule is then more likely. But if it was bad in optimizing a desired activity profile, then I want to make that less likely. So I want to make sampling a similar molecule now less likely according to my policy. And now I keep repeating the same process. So I keep generating molecules according to my policy, where policy is just those probability distributions. And then I keep iterating and I keep refining my policy based on the rewards that I'm getting. And at the end, hopefully I end up with an optimal policy that is able to generate active molecules. Yeah, so that's basically what I do. And then to answer the grand question of our talk, so can AI design drugs in general? So coming back to the main problem that we started off with. Well, the answer is that sort of, it sort of can. So actually, Exscientia, in collaboration with another company, has recently produced a drug that, sorry, not recently has produced a drug, but has recently, the drug candidate that they have produced have recently entered human clinical trials, which has never happened before. That is a first drug candidate designed by AI or with help of AI that is entering human clinical trials. So that's pretty cool. But the thing is that when we say that AI have designed a certain drug, it actually oftentimes isn't as futuristic as it might seem, as in it's not a full stack pipeline. AI is not doing every single step of the process. It is very much still a process that is monitored by humans, and we design it, and AI is just like helping us make some steps of the pipeline faster and is doing some of the searches for us. But there's still a lot of stages of the process that stay the same or stay monitored by humans. And I think in my personal opinion, like that's probably not going to change anytime soon. So I think the way that we're going to try to apply AI to the drug design process, that we're going to be applying it to optimize it further and further and make it faster and faster. But it's going to be very difficult, I think, to design to be designing drugs with AI only. Nonetheless, it is definitely an exciting future to look forward to. And just last thing that I want to mention, and then we can move on to the questions. If any of this sounded interesting to you, and maybe you think that this would be something that maybe you would want to work on, then you can check out open positions that we have at Exscientia, because we're growing quite quickly. So Exscientia is looking into hiring quite a lot of people. So yeah, if you're interested, check out our website. I've put a link right here. Yeah, with that, I think we can move on to the questions.
Hello, yes, sorry about that. Well, first of all, thank you so much for such an amazing talk. I mean, wow, what a world we live in. I mean, it was just absolutely incredible. So yeah, we do have a number of questions. So I'll get started with the first one, which is, have you at Exscientia been working on anything related to COVID-19, or are you not allowed to disclose that sort of thing?
I don't think I'm allowed to disclose a lot about it, but I think there have been some press releases. So I think I'm allowed to say that Exscientia is looking into working on COVID-19, but it definitely would not be any work in terms of designing a vaccine, because we design drugs. So it would be a slightly different approach. And it might be more of like a preventative work in the future. So if there was a risk of a global pandemic in the future, maybe related to a different virus, then maybe we would want to develop some standardized approaches to tackle that, so that it doesn't escalate as quickly as COVID-19 did this year. I think that's all I'm allowed to say.
Right. Yeah. No, I completely understand. Thank you. I'm going to take a question from the chat now, which is, what language do you mainly use? How much of the material that you've discussed was taught as part of your degree? And so to paraphrase, how useful did you find your degree?
Okay, there's a lot of stuff to unravel in this question. So in terms of the language that we use, we primarily use Python. How useful was my degree for what I do now? So I think the most useful components were probably the projects that you can do. And I would definitely say that the final 2 years of my degree were probably more relevant to what I do now than the first two. But then I also think it's important to like develop good foundations so that you can build a solid understanding of the concepts that you're working on. But yeah, just in terms of like the domain, I think there's definitely some courses. So for me, like all the courses that had like reinforcement learning elements in them, I think were pretty useful. But also, I think it's really important to like develop good sort of like work practices. So, and I don't mean it in like an employee sort of way. I mean it as like working on your own personal project sort of way. So you know how to manage your time well. And I think for that, it's pretty useful to do all sorts of different projects at the university, both group projects that allow you to interact with your teammates, and personal projects as well. Yeah, because there's always ups and downs, especially in research. And there's moments where you get stuck and you just don't know how to push a certain approach further, and maybe you don't know how to implement something. So I think it's good to have those sort of experiences at when you're still in uni, so that it's like a very controlled environment and there's still a lot of people who you can ask for help. So then when you move on and you either, like go on the academic path and pursue a PhD, then you know how to tackle a much larger project, or likewise in the industry, you just know a bit better what works for you and what doesn't, and how to keep yourself motivated. So yeah, I think that would be my answer.
Awesome, thank you. The next question from the chat is, can you be more specific on sustainability and profitability of this tech at its peak? Not really sure what it means, but that's the question.
Right. So as and so I guess it's a tricky question, because so the phases of the drug design process that we work on, they have very little to do with actually producing drugs. That's a much, as in, don't get me wrong, I mean physically producing drugs, where I think the sustainability factor has a large play. So for instance, if you're, like if your drug is already approved and you're trying to get it to the market to be able to sell it to people, then there's a large discussion around how do you actually produce it, what sort of packaging it's going to be in. I think that has a lot more impact of the environment in terms of like the sustainability sort of way than the intellectual process has. So yeah, actually designing, at least the way I understand the question, I think there's not that many concerns with sustainability in the particular problems that we work on. But yeah, if there's any follow-ups on this question, maybe a bit of sort of like explanation around what exactly they wanted me to cover, then I'll be happy to go back. But yeah, I'm not entirely sure if that's the answer that you are looking for.
Sure, yeah, I'll keep you updated if the commenter says anything else about it. So, what's a typical day in the life for you look like? With
Well,
regards to work, that is.
sorry.
With regards to work, that is.
Yeah. So obviously it has changed during COVID and the lockdown. I am currently still working remotely from home. But so maybe that answer should have sort of like two parts to it. The first one is when we were still in the office, and then the second one is during the remote work. Yeah, so usually, so the working hours that we have is the standard 9 to 5. So I would usually just get into the office at 9. I would usually have breakfast in the office, and then I would start work. There's not too many meetings that you have during the day as part of like the AI team. I think the chemists in our company or the biologists, that they tend to have a lot more meetings. But yeah, we sort of do our own thing. We just like within the group, there's a lot of communication, but a lot of it is pretty informal. Like you don't really have to, which I actually really like. You don't really have to like schedule a meeting with someone because most of them are sitting next to you. So if you have a question, you can just like pop a question to them. And yeah, so during the day, you work on your project and you're trying to make progress. On some projects you have teammates, so then a lot of stuff you'll be discussing with them. Some projects are a bit more individual, and every maybe weekly you have some sort of catch-ups to make sure that you're making good progress. Yeah, and then I would leave the office usually like at around, I'd usually stay a bit longer, so I would leave at around 6 maybe. Oh, you have lunches in the office. That was also really nice. And all sorts of snacks and really good coffee. So definitely something that I miss while working at home. And yeah, then I would just get back home. So that was sort of like the life in the office. But right now it's obviously a bit different. So I still try to stick to more or less the same hours, and obviously all of the meetings happen online. So it's a bit different, and it actually took me yeah, there was like a learning curve into working at home. I really liked sort of like changing my environment and going to a different place where you can feel like you're in a work zone now. But yeah, I think it's a lot better now, and it's like it's still like the same computer that you work on, and yeah, that the time hasn't changed as much, and like sort of like the the types of tasks that you're working on as much as the environment. I think the biggest difference between like working in the office and working at home is just like the random interactions that you don't get to have, or you actually need to like set up a meeting with someone if you want to discuss something, and you can't just like, like turn over and ask them a question. But yeah, other than that, it's pretty similar in terms of like the working hours and the setup of the meetings and the projects that you work on. So yeah, that's my day at work.
Awesome. The person earlier who asked about the sustainability and profitability thing rephrased their question. So the question now states, I wanted to know how disruptive this tech would be in years to come.
Right. So I think that is a really good question. And I think it's a type of question that you often see with applying AI to all sorts of different domains. And it's this sort of attitude of like AI being the enemy slightly, or at least I sometimes think that. But yeah, I think so. I think in this particular case, it in terms of like how disruptive it would be, I don't think the effect of it is going to be any different than for the human design drugs, because I don't think the clinical trials, that process, I don't think that's going to change anytime soon. So whether it is a drug suggested by a researcher or a medicinal chemist, or if it's a drug suggested by AI, they both are generating or suggesting just drug candidates. And those candidates still need to go through all the phases of testing. So you still need to make sure that the drug is effective, and you still need to do the clinical trials to make sure that it's actually doing its job and it's not affecting any other processes in our bodies. And, like that, everything stays the same. So I think, or maybe I hope that the technology that we're developing is not going to be disruptive. I hope that answers your question.
I hope that answers his question as well. I think we just have time for one more question today, and there isn't anything else in the chat, so I'll ask mine, which is, if you could go back in time to say when you were 20 years old in your penultimate year of your undergrad, I think this was at Caltech for you, and you're on the brink of your career and indeed your life for that matter, and you could give the younger Ola any piece of advice, what would it be?
That's a really good question. I think I think for me, it would be something along the lines of like, don't take everything super seriously. At least I used to have this mentality, or probably still have a bit of it, that if I have a plan, for example, of like getting into a certain university or then pursuing a certain career path, and if for whatever reason something wouldn't work out, then I would get like or I would be like very goal oriented in that sense. So I would try to like always pursue the path that I have imagined. And the thing is that I think that no matter what happens, like even if something fails on the way, like I think you're going to sort it out and find maybe a slightly different path, but something that's ultimately also going to make you happy. So I guess the advice would be to not think in this like grand terms that like it's either all or nothing. There's a whole bunch of things in the middle, and I think like we as humans are pretty good at like figuring out how to make life work for us. So yeah, I think that would be the advice. Like even if you fail, it's still okay.
I think that's some phenomenal advice. I'm just going to check the chat to see that we don't have any other questions. It seems that you did adequately answer the other gentleman's question. So that brings us to the end of our time. So thank you so much for coming. I mean, this was a phenomenal talk. I think a lot of people enjoyed it. and it is going to be available for replay for if in case you want to go back and watch some part of it or you missed it. So yeah, thank you so much. Would you like to make any closing remarks, perhaps?
No, I think that's it. I would just like to thank everyone who decided to join and listen to me. Thank you for staying till the end. Yeah, thank you for giving me the opportunity to present and yeah, give this talk. I really appreciate it, and it's been a really good experience. So thank you, everyone.
Glad to hear it. It's our pleasure. So if you'd like to end the live stream, if you just go into Zoom, click on more at the bottom, and then stop live stream, and that'll end the live stream.
Oh.
I'll tell you when it starts.
So
So there's more at the bottom, like